Hi everyone, my name is Hoon Cho. I'm from Yale University, and I'm one of the co-organizers of this workshop as well. I just want to start by saying how much I appreciate being part of this workshop, and I'm looking forward to many of the conversations and all the talks we'll have throughout the program. So in this talk, what I'll do is share some basics of genomics and talk a little bit about applications of DP in genomics to hopefully provide a very brief primer on these concepts for those of you who aren't too familiar with genomics as a field. I understand that, yeah? Okay. Perfect, thank you. Where was I? So I understand that we're a little bit behind schedule, so I'm gonna try to keep things briefer than I planned. So I'm gonna start by quickly defining genomic data. What is it? What does it look like? So a quick biology 101, just a couple slides. So the human genome includes 23 pairs of chromosomes, right? And each chromosome is a long DNA sequence consisting of 4 nucleotides, ACGTs. You're all probably familiar with this. And there are 3 billion bases in total in the human genome. In genomics, we often care mostly about the positions in the genome that differ across individuals. So these are what are called genomic variants. It's typically around 0.1% of the genome that's different between any given pair of individuals. There are different types of genetic variants, with single nucleotide polymorphisms, or SNPs, being one of the most common types. So it's single letter differences in the genome. And when we think about genomic data, it's often represented as these binary sequences, and this is because we can encode things, the genetic variants, relative to a common shared human genome reference. So the sites that are identical to the reference genome, it would be encoded as zeros, and then sites that are different would be encoded as ones. So then each individual has 2 binary sequences because everyone has pairs of each of the chromosomes. So this is what the genomic data typically looks like when we talk about genomics. One important thing I want to point out is that genomics is broader than just the DNA sequences. So there are these other types of omics data, as Lee mentioned, including things like epigenetic data, which are chemical modifications to the genome. And then there are also measurements of other types of biomolecules in our bodies, so things like mRNA molecules, which are produced from the DNA. So measuring those give us a notion of how active each gene is in the genome. So that's an area called transcriptomics. There's also measurements of protein levels, and we can also study that. So all these things we have experimental technologies to measure and study. So in our context, the use of these data and the application of DP to these data types are all within scope of what we are trying to address here. So given these different types of data, one of the major goals of biomedical research is to link all these molecular types of information to health-related information about individuals. So what's often done, as you may be aware, we collect these genomics or omics data linked with health records across many individuals, and so we can try to study the link between these 2 different data types. And what that allows us to do is first we can try to look at the associations and learn about the biological mechanisms behind human disease and human health, and also use that knowledge to build predictive models that can help us with clinical decision support. So when we try to leverage genomic data for these different purposes, we run into different privacy risks, as you know, so that's not a surprise. But here I wanted to point out some of the unique aspects of genomic privacy. So the first thing, as Lee mentioned, again, genetic sequence is highly unique. So there are millions of sites that are unique to each individual. So what that means is that it increases the risk of membership inference when an adversary is combining this data from a target individual with observed data that's released from the private cohort. And second, there's also the risk of genealogical re-identification. And what this means is that we start from an anonymized genetic profile, and using the fact that these genetic sequences are shared between biological relatives, we can query the sequence against public genealogy databases to find the relatives and then try to triangulate and identify who that person is behind that genetic profile. So this is a risk that's kind of unique to genetic data. And lastly, I want to mention data linkage risks. So since genetic sequences are potent identifiers, it can be used to link the same sample across different data sets. And because the other omics data types that I mentioned, they're correlated with genetic information, one can often use those data to infer the genetic information of the same individual. And this allows us to sometimes link across different data types as well. So in the next few slides, I'll talk about some of the key use cases of DP relating to genomic data. This first example is about secure release of study results. So here we're hoping to publicly share aggregate statistics or train model parameters from private cohorts and release them under DP guarantees. One example that I'm showing you here is related to genome-wide association studies, or GWAS, where we're taking genome-wide measures of correlation between genetic variants and different health-related traits of interest, and then we're interested in releasing that with the public for secondary analyses. But of course, that introduces privacy risks, so how can we do that in a privacy-preserving manner? And this is actually an actively studied problem over the years, and existing methods have addressed different types of statistics that we want to release, different analysis models. And I wanted to give a quick plug to our recent work, which was presented at Recom just last week, where we addressed the common issue of not being able to release millions of statistics at the same time by optimizing the randomization function that we apply. I'd be happy to talk more about that offline. This next use case I wanted to point out is queryable databases. So here the high-level problem is we have all these large biomedical databases. We want to open them up somehow to allow researchers to interact with these databases to obtain answers to different types of analysis queries that they may have. And there are different types of settings where this can be useful. So for example, we can have clinical databases where the users are interested in defining a study cohort based on some match criteria. There are also these services called genomic data beacons where we're interested in having users look up particular rare mutations of interest to help with clinical diagnosis. And there's some of these other tasks where, and these are existing real-world services as well, where we leverage reference panels, so a collection of genomes collected from many individuals to facilitate different types of genome analysis tasks. And lastly, this was mentioned before in previous presentations, there's synthetic data generation, and this is emerging as a key application of DP in this domain as well. And of course, we're aiming to generate shareable data sets with some privacy guarantees that also mimic the statistical properties of real data that we're interested in. And this is widely useful for different purposes. So it helps with community benchmarking on different method development tasks. It helps to prototype research or software tools very quickly. And it also helps with increasing the training data size for different problems. And related to this, I wanted to point out this recent development. There's been this recent challenge that was organized at ISMB on synthetic omics data generation. So this is interesting because they're focusing on generating cancer transcriptomic data, so gene expression data, one of the omics data that I mentioned earlier. They looked at a wide range of different methodologies, including classical models and machine learning models, with or without DP, and then compare the utility according to different metrics that they introduced that are specific to this problem setting. And there's this nice summary article on their findings from last year's competition in case anyone's interested in looking at this. So I'll end by sharing some thoughts on why, in many cases, it can be challenging to develop practical DP methods for genomic data. So this is taking a more data and modeling related perspectives, complementing other perspectives that Rachel and Lee shared earlier related to governance and user engagement and so on. So I think one of the key challenges is that genomic data is very high dimensional, again, millions of variables per person. So how do we cope with that and also address limited sample sizes typically in these biomedical studies to have something that gets a reasonable balance between privacy and utility. And then the second challenge is that in biology, oftentimes rare patterns in the data sets are the most interesting ones. So once we add DP, we might be losing those signals. So how do we retain those signals for these use cases? And then there are these other interesting method development opportunities, including things like methods to account for the inherent noise in the experimental data sets that are pretty common, and also the ability to support more higher complexity AI models that are being introduced in this field lately. So with that, I'll pass it back to Lee to start our next session. Thank you.